Welcome to Simplified UPSC by PPP, where we listen, learn, and grow together. Detailed lecture notes in English and Hindi, plus practice questions for prelims and mains, only on our Telegram channel. Link in description. Like and subscribe for more.
Before we jump into the deep dive today, just a quick and humble request. If you find what we do valuable, and if you're able, please consider subscribing and maybe even joining the paid membership of the Simplified UPSC by PPP YouTube channel.
It genuinely helps us keep creating this kind of, well, super useful content for you.
And a huge, huge shout out and our deepest appreciation to all of our existing members.
Yes, truly. Your support is absolutely what keeps this going. And of course, don't forget to like this one and share a comment. It helps more than you know.
Okay, let's unpack this. Our mission today is a very tailored deep dive. It's for you, the learner, who's focused on rapid but still really rigorous preparation for exams like the UPSC.
Exactly. We're synthesizing the really high-yield concepts from the NCERT Class 11 Biology Foundation. We're focusing on Chapter 3, and we're linking that theory straight to insights from previous year questions we've got in our source material.
And what's so fascinating to me is just how these foundational concepts, from defining life itself to understanding biological cycles, are tested.
They come up again and again. So we're going to distill all that dense info into clear, interconnected nuggets. We want to make sure you not only know the facts, but really understand why they matter.
So let's start at the very, very beginning, the fundamental question. What makes something living? Our source material lays out this whole list, growth, reproduction, ability to sense the environment, metabolism.
It's a long list.
Right. For someone studying, what's the hierarchy here? What are the concepts you just have to know?
That's the critical distinction right there. We have to separate the general characteristics, the visible features, from the actual, the defining properties of life.
Okay.
Take growth, for example. A mountain can grow, right? By accumulating matter on its surface. But we don't call a mountain alive.
And reproduction, that seems like a sure thing.
It's key, but it's not universal to every single living thing. Just think about infertile worker bees. They're definitely alive.
So what is the gold standard then? What's non-negotiable?
It's really twofold. First up, metabolism. We're talking about the thousands of chemical reactions happening all at once inside every single cell of every living organism.
But you could run one of those reactions in a test tube. An in vitro reaction.
Precisely. And that single reaction happening outside a system doesn't make the test tube alive, does it?
Which brings us to the second defining feature, the system that contains all that activity.
Exactly. Metabolism only becomes a defining property when all those thousands of organized reactions are contained within this structured system. We call it cellular organization. That's the defining feature. Nothing non-living has it.
Okay. And there's one more, right? The most obvious one.
The most obvious and maybe the most technically complicated is consciousness, the ability to sense and respond. And specifically for humans, it's that next level, self-consciousness.
Right, so metabolism and cellular organization are the definitive properties. They're exclusive to life. And all that diversity of life, it needs to be organized. Let's touch on taxonomy, the hierarchy from species all the way up to kingdom.
And there's a crucial pattern here that gets tested a lot. As you move up that ladder from species to kingdom, the number of common characteristics shared between the members, it continually decreases.
So the closer you are to the species level, the more things they have in common.
That's it. It's an inverse relationship.
And this classification, it's not just abstract theory. It links directly to really practical questions. Let's connect this to a previous year question about biofertilizers. There's a question asking which organisms are used as biofertilizers, and the options included agaricus, nostoc, and spirogyra.
A perfect example, this is where you have to synthesize function with classification, and the right answer there is nostoc.
Why?
Well, because nostoc is classified as a cyanobacterium or a blue-green alga. It's a photosynthetic autotroph, and here's the kicker. It's known to fix atmospheric nitrogen. It does this in these special thick-walled cells called heterocysts.
And agaricus.
Agaricus is a fungus. It's a mushroom, so it's a heterotrophic decomposer, a completely different role.
So just knowing the basic classification lets you eliminate the wrong answers based on their fundamental function.
Yeah, exactly.
Okay, if metabolism defines life, we have to talk about the biggest metabolic process on the planet. Photosynthesis. It's the engine of the plant kingdom.
Right, and the history is quite interesting. You had Priestley showing that plants somehow restore air that's been, damaged by a burning candle or an animal breathing.
And then Ingenuus built on that.
He did. He proved that sunlight is absolutely essential, and more specifically, that oxygen is only released by the green parts of a plant and only when they're in bright light.
And this led to our understanding of the two major stages.
The light and dark reactions.
Let's break that down. The light reactions. They happen in the thylakoids, those little stacked discs inside the chloroplasts.
Right, the grana. And their whole purpose is just to generate the energy currency.
ATP and ADPH.
Exactly. And the process of making ATP using light is called photophosphorylation. This is the energy capture phase.
And the dark reactions, the biosynthetic phase.
That happens in the stroma, the fluid-filled space of the chloroplast. It uses that ATP and NADPH from the light reactions to fix CO2 and actually build the sugars, the food.
So the dark reaction doesn't actually need to happen in the dark.
Not at all. A common misconception. It just doesn't directly depend on light, but it is completely reliant on the products of the light reaction. It was Melvin Calvin's work with C14 that showed the first product in most plants is a 3-carbon acid, PGA, hence the C3 pathway.
Which brings us straight to a key distinction, one that's almost guaranteed to show up on exams. C3 versus C4 plants.
The C4 pathway, or the Hatch and Slack pathway, is just a brilliant evolutionary adaptation.
So what problem?
To a really wasteful process called photorespiration. It happens in hot, dry conditions when the main C3 enzyme rubisco accidentally binds to oxygen instead of CO2. It's a huge loss of efficiency.
So C4 plants found a way around this wastefulness.
They use spatial separation. They separate the initial capture of CO2 from the final step of making sugar. In a C4 plant, the first CO2 acceptor is a 3-carbon molecule called phosphenol pyruvate, or PEP.
And this happens in a different place.
It happens in the outer mesophyll cells using an enzyme called PEP carboxylase, or pepcase. And the key thing about pepcase is that it has basically zero affinity for oxygen. So photorespiration is impossible at this first step.
Okay, so the CO2 is captured. Then where does the sugar production, the Calvin cycle, actually happen?
The resulting 4-carbon acid gets shipped, so to speak, to an inner ring of cells, the bundle sheath cells.
That special anatomy, what's it called?
Kranz anatomy. These cells have thick walls, very few spaces between them, and this is where the Calvin cycle happens. The bundle sheath cells are packed with rubisco, but they have almost no pepcase.
So the genius of it is that the first step acts like a super efficient pump. It just shoves massive amounts of CO2 into those bundle sheath cells, so rubisco has no choice but to bind with CO2, not oxygen.
That's it, exactly. The Calvin pathway itself is common to both C3 and C4 plants. But the location and that high concentration of CO2 around rubisco is what makes C4 plants so much more productive in high heat and sun.
Now let's pivot from making energy to, well, synthesizing nutrients.
Yeah.
Specifically, biological nitrogen fixation. We know atmospheric nitrogen, N2, is basically inert. So this biological conversion to usable ammonia is vital.
It's absolutely vital. And you have to remember this process is performed exclusively by prokaryotes. We call them N2 fixers.
And they come in a few different flavors.
They do. You have free-living aerobes like Azotobacter, free-living anaerobes like Rhodospirillum, and then the cyanobacteria like Anabaena and Nostoc, which, hey, we already mentioned as biofertilizers.
The triple bond in N2 is incredibly strong. So what's the challenge for these organisms?
It's a two-part challenge. Energy and oxygen. That stable bond is broken by an enzyme called nitrogenase, and this enzyme is extremely sensitive to oxygen. It gets destroyed by it.
So the whole process has to happen in an anaerobic and oxygen-free environment?
It has to. And then there's the energy problem.
What's the cost?
It is staggering. It takes 8 ATP molecules to produce a single molecule of ammonia.
8 ATP for 1 NH3. Wow. That really shows you how stable that N2 molecule
It
is.
really does. It's a massive energy investment.
So how do the symbiotic fixers, like in lagoon root nodules, solve that oxygen problem? I mean, the root needs oxygen to live.
They have a very clever defense mechanism inside the nodule. The plant produces an oxygen scavenger molecule called Leghemoglobin. It binds really tightly to any free oxygen, basically mopping it up. This creates that necessary anaerobic microenvironment to protect the nitrogenase enzyme so it can do its job.
Just a quick note on structure before we move on to life cycles. The plant's integrity, that's the cell wall.
Right, providing shape, protection, made of cellulose, hemicellulose, pectins, and so on.
And that ties back to how plants develop. Their cell differentiation is described as open.
Which means they show plasticity. They can change their developmental path depending on the environment, a crucial adaptation for something that's, stuck in one spot.
Okay, finally, let's get to alternation of generation. This is the core reproductive strategy for the whole plant kingdom.
Right. It's the cyclical shift between two distinct phases.
The diploid phase and the haploid phase.
The diploid sporophytic phase, which produces spores, and the haploid gametophytic phase, which produces gametes. And how dominant and independent each of these phases is, well, that defines the three major life cycle patterns we see in plants.
Let's run through those three patterns, maybe with an eye on the evolutionary trend.
Okay, first up is the haplontic life cycle. Here, the dominant, free-living, photosynthetic phase is the haploid gametophyte. The diploid sporophyte is basically just the single-celled zygote.
And we see that in
Many of the more primitive algae, like Spirogyra.
and the opposite end of the spectrum, the most advanced pattern.
That would be the diplontic life cycle. Here, the script is completely flipped. The dominant photosynthetic free-living phase is the diploid sporophyte. We see this in the alga fucus, but significantly, it's followed by all seed-bearing plants, the gymnosperms and the angiosperms.
And the gametophyte in those plants is tiny, right?
Highly reduced. It's dependent on and contained entirely within the sporophyte.
So what's the middle ground, the transition?
That's the haplo-diplontic pattern. Here, both phases are multicellular, but one is usually more dominant than the other. This is the intermediate condition you see in bryophytes and pteridophytes. It really shows that clear evolutionary march towards sporophyte dominance.
So let's synthesize this. What does this all mean for you in your preparation? We've gone from the very definition of life to how taxonomy links to real PYQs on biofertilizers. We've covered the subtle but really profound differences between C3 and C4 photosynthesis,
Focusing on that adaptive advantage of Kranz anatomy.
and the huge energy cost of nitrogen fixation. I think the key nuggets are understanding those defining features of life, metabolism, and cellular organization, and then the contrasting energy pathways, and finally, that evolutionary trend toward the diplontic life cycle in the most advanced plants.
And if we tie all that together, thinking about the big picture of evolution, we see that the diplontic life cycle, that dominant sporophyte, became the winning strategy for all seed plants. Which leaves us with an important question for you to think about. Considering that the evolution of the seed itself required the female gametophyte to be retained inside the ovule, what was the key adaptive advantage that this complex diploid dominant life cycle provided that made it so universally successful for these most evolved plant groups?
An excellent thought to leave us with. The why of evolution is so often the key to the what on the exam. And that is all the time we have for this deep dive.
Once again, a huge thank you to all our existing paid members of the Simplified UPSC by PPP YouTube channel. We are so grateful for your support.
And if you found this valuable and you're in a position to, please do consider subscribing and joining the membership. It helps us continue to create this content for you. Don't forget to like and leave a valuable comment below. Until next time.
Thanks for watching. Follow us on Telegram and Instagram. Don't forget to like, share, and subscribe.